Right, so I think we'll get started. Like, thank you all for coming and showing up. I'm actually super excited to introduce our speaker for the day, for tonight. It's kind of dark on the outside. Dr. Kevin Mitchell from Trinity College Dublin, and he's an associate professor in neuroscience and genetics. I was told that that's the easiest to say. And so Kevin, let's give some credit to our supporters. He is here because ELTE and TCD are both in this Charm EU program, and Charm EU was nice enough to fund Kevin Searle, but he's really here because I think he's like one of the more original thinkers of our time if it comes to biology. And if you read his books or read his papers, you can always find something that's really exciting and interesting. And as you might know already, he is going to give two talks. Like, one is today about the genomic code, one of his latest papers and latest ideals, maybe the beginning of a new book, I understand. And tomorrow there will be at the Hungarian Academy of Sciences another talk, which is about a very hot topic, the free will, whether there is or there isn't. I don't want to give.
There is.
Okay, so today we are going to hear about some developmental genetics aspects of Kevin's work and his thinking. Some of it can be found in his book called Innate. I really recommend that. It was like a huge revelation when I read it, and also in a more recent Trends in Genetics paper, which is titled The Genomic Code. How our genome essentially encodes information. And without further ado, Kevin, please.
Thank you so much. Thanks for the invitation. Thank you all for coming. It's a pleasure to be here. Thanks to Charm EU for supporting it. And so what I'm going to do is, this is really just a really conceptual talk. So I'm not going to be presenting any original data. This is really an attempt at a synthesis for thinking about how we should think about how the genome encodes the form of the organism and the nature of the organism as well, though I won't really talk about that too much. This is one of the most sort of fundamental core observations of biology, is that like begets like. And it's so fundamental that we don't pay a lot of attention to it, actually. We just sort of almost don't notice it. But how cats have kittens and dogs have puppies is like a fundamental question in developmental biology and genetics and evolution. And of course, people have thought about how that could be in many different ways. And going back to antiquity, one of the ways that people thought about it was this idea of preformationism, that actually inside the little, sometimes it was the fertilized egg, but often it was just the sperm or just the egg. There must be a little preformed thing already there. There was this idea that you couldn't get real new stuff if it wasn't, it had to already be there. And so, there were lots of people in, the ancient Greeks who believed that. Aristotle was not one of them, and he actually looked. So, novel idea. Look inside and see what you see. And when he looked in developing chick embryos and so on, he didn't see a little chick with all the little preformed parts. So it didn't make much sense. And of course, now we have much better microscopes. You can just look inside a fertilized egg and you don't see a little thing, right? So you look in a cat zygote and a dog zygote, you obviously don't see a little dog or cat, right? But something in there is carrying some information about the form that emerges. And so Aristotle proposed this idea, which came to be called epigenesis, which is just the notion that you do get a gradual emergence of form through development, through the processes of development that isn't there at the start, but something's there. Right? So he called it the eidos, or the idea. The idea of the organism is somehow there in some physical material. Right? So he already had what we would now call the idea of genetic material, even though he didn't know what it was made of. And he also thought it was just in the sperm and not in the egg. So didn't get it quite right, but he was onto something, because obviously something has to be different up here at just the fertilized egg stage of all these things that determines what they develop into. So this idea then is obviously old in that sort of notion that there's some physical stuff that encodes the idea of the organism. And that's the question that I'm interested in, is thinking about how we should think about that. right? Of course, it was shown that DNA is the genetic material in these sort of classic experiments with bacteria and in the cloning of sheep and lots of other things since then, where the form that emerges depends on the DNA in the nucleus that's transferred, not the host. Okay. So now that doesn't mean that DNA is doing anything by itself, because it's incredibly inert, stable molecule. That's one of its main functions, is to be super, super stable, right? So the idea that there's information in the genome does not mean that all of the information that's required to make the embryo is in the genome. In fact, the genome doesn't have to encode loads of things because the environment provides them. Either like the physical environment of the egg provides it, or the womb, or wherever that's developing, but also just like physics provides a lot of the information for how development happens, and the genome doesn't have to encode it, right? But still, even with that, right, we don't have to say genetics, the genes does everything, but it clearly does something special, right? Something different from the other stuff in the environment. So something about the dog DNA or cat DNA determines what kind of organism it develops into, and that's the question that I want to get at from a kind of a philosophical point of view, is just how should we think about that, right? How is the form of the organism represented in the genome in some way? And again, you can have a wrong view of this, a kind of a preformationist view where the representation is like not in the egg, but when you look in the genome, you somehow see a little doggy, and you don't, right? So how is it actually encoded there? And people over the years have used various kinds of metaphors. One of the most common ones is to say a blueprint, right? Your DNA is the blueprint for the organism. And it has the benefit that it's familiar, right? So a good metaphor latches onto an idea that people already have, and it makes the unfamiliar familiar. The problem is it's completely wrong and absolutely doesn't work at all, actually, once you start thinking about it, because, of course, a blueprint, the bits of the blueprint correspond directly to bits of the output, and that's clearly not how the genome works. This was, at least some of the first people to talk about this were Jacob and Monod and their famous paper that was looking at gene regulation of the lac operon in E. coli. And they were thinking about how to think about what the genome is doing, and one of their arguments was that the genome has these independent blueprints for building proteins. And their blueprint is perfectly adequate. It's absolutely a reasonable way to talk about it, because there is a kind of a direct isomorphism between the information in the genome that's coding for a protein and the protein, right? It's a very direct, linear, one-to-one mapping between the codons and the amino acid and the protein that emerges. But clearly that's not enough, right? Deciphering the genetic code, which led to understanding of how these codons map to amino acids, is not the secret of life. And I think a lot of people think when the genetic code got mapped, it's like, yeah, there, now we're done. But we're not done, because of course, the secret of making dogs or cats is like how these proteins are all used, right? How they're actually expressed. It's a question of gene regulation, not the proteins themselves. And Jacob and Monod recognized that as well, that it was coordination of those genes that was the really important thing. And so the genome contains not only a series of blueprints, but a coordinated program of protein synthesis and the means to control its execution. And that was the first mention of this idea of a program, right? So rather than a blueprint, which is this static thing with this one-to-one mapping, now there's a program, which sounds better, right? Because it's a dynamic sort of a process. The problem with the word program here is it has a couple of meanings, right? One meaning is just a series of steps, something that happens through a series of steps, right? Like the program for a music recital. It's like, okay, they're going to play this, then this, and this, and this in this order, right? That's a program, that's fine. And you can think of development like that. It has a series of steps that happen in an order. Clearly that program, that developmental program, is somehow encoded in the genome. But it's not encoded as a program in the way we think of like a computer program. So it's not encoded like an algorithm where you can look to bits of the genome, and this bit of the genome says, now do cell proliferation in the kidney, and this bit says, now make a tubule, and now make a whatever, right? It's just not a program in that way. And so the word has an unfortunate connotation in modern terminology, because we think of computer programs in that algorithmic way. And it makes it sound very deterministic, right? Which is one of the problems also with the blueprint metaphor. It's just really like whatever the information is there in the genome, that's what's going to be the outcome. And it's not like that. It's not deterministic. So a program is okay for describing the fact that development goes through a series of stages. It's not okay if it implies this kind of algorithmic thing. Another metaphor, and this is one I've used myself, is a recipe. And I think this is a bit better than just a program. The idea is that you with a recipe, you put some stuff together in a certain way, and then stuff happens, right? But where the happening is not part of the recipe. The happening is like the physics of eggs and flour and heat and whatever stuff. This sort of self-organizing physics in the material. What the recipe does is organize it in a certain way and set it off, right? And that's a little closer to, I think, the way we should think about embryonic development. The problem, of course, is that this recipe here has a baker, right? And there is no baker outside the thing making it. The thing has to make itself. So that metaphor falls down a bit as well. So we have these sort of three major ones that have been used and still are very widely used. The blueprint, the problems with it is like bits of the blueprint correspond to bits of the animal, which is obviously not right. The program bit has the same problem except with processes as opposed to anatomical parts. And then the recipe has the issue that the thing has to bake itself, and it's not obvious how that actually happens. Although it has the benefit that it makes use of these non-encoded physical and chemical properties that are just in the world, right? Don't have to be encoded. Another one that is interesting came from Erwin Schrödinger, who gave a series of lectures actually in Trinity College in 1943. So he had moved from Germany to Dublin to set up the Dublin Institute for Advanced Studies. And one of his roles there was to give these public lectures every year on various topics. And at some point, he turned his attention to biology and was thinking like a physicist that biological beings, basically what they are is organized processes of matter that keep themselves organized despite the second law of thermodynamics that says they should go to equilibrium with their environment, right? So they don't become one with the universe. They keep themselves in a pattern, and what he said is that pattern has to be specified. It's not just any pattern, right? It's a pattern of a dog or a cat or an elephant or whatever it is. So it was known that the chromosomes probably contain the genetic material, and for him, that had to contain some kind of what he called a code script. So he was thinking about information in a thermodynamics sense, where the sort of the physical nature of the encoding material had to be stable, but it also had to have enough variation in it that it could encode some information. And one of the interesting things was that he realized it's not enough to encode what you could call the architect's plan, the blueprint of what it should look like at the end. You have to encode how to build it. And those are not the same thing, right? So the builder's craft is different from the architect's plan. But still, code script is a bit loose, right? It's just not exactly clear what that really means. It's a bit vague. So in thinking about this, you can think about the job of the genome, right? What's the role of the genome is to allow recapitulation of the pattern of the organism from a highly, highly compressed set of instructions. So if you think about the amount of information that it would take to do like the Star Trek replicator beam me up kind of a thing, right? That replicates every atom position, right? Just hundreds and hundreds of orders of magnitude more than the information that can be encoded in the genome. So that all of this information here emerges from this massively compressed set of instructions in the genome. And that gave myself and my colleague, Nick Cheney, an idea, which is that there's an analogy here to machine learning systems. So a lot of machine learning systems that you guys will be familiar with, the way that they learn things, they learn to recognize, say, cats and dogs, right, is by seeing loads and loads of pictures of cats and dogs and compressing this abstract representation of dogginess or cattiness from all of those examples without holding on to all of the details. In fact, it's the compression that allows them to abstract the larger kind of idea, and then they can reconstruct it, at least the ones that have a generative kind of a model. I'll show you an example of that. So as Mante said, this is from a paper that we published this year, I guess, which proposes this idea. And this was one of the examples of a type of machine learning system that we drew on as an analogy here. So this is a system where, so this is a system with neural nets. Each of these is an artificial neuron. They're arranged in layers, and the layers have these more or fewer elements in them that enforces convergence or divergence. And they're trained on lots of things. This could be like images of, say, horses, right? So trained on loads and loads of images of horses, and then they develop this compressed representation in the sort of middle layer here, and then at the same time, they develop a means to decompress it to make a new instance, right? So if you've ever used like Midjourney or DALL-E or something like that to draw something, that's what they're doing. Okay? So here's one. Here's a nice horsey that I generated from Midjourney, I think, that clearly has an abstract, compressed model of horsiness that doesn't depend in any of the details, right? And it's generating really a new thing that has never been seen before. So the idea is that our genome is something like this compressed representation, and this has some properties that are really interesting and that can be formalized mathematically in ways that I think can be useful for thinking about genetics. So first of all, it's really distributed. There's only 2 nodes here. Obviously, the real thing has hundreds and hundreds of nodes, right? And it's the nature of the encoding there is actually largely uninterpretable. So when people train these things, and then they look inside there to see, okay, which of these nodes correspond to bits of the outcome, they can't find it, right? There's no, it's really hard to decipher the logic of it. And the reason is it's hugely nonlinear. Right? The relationship between bits of the encoding representation and bits of the output is not one-to-one. It's massively nonlinear and distributed. And so they refer to that as a space of latent variables, where the variables in that case are the weights between all of these neurons and the inputs and outputs. Okay. So the idea is that the analogy here is that this is like our genome, and that evolution is basically doing this job. So through generations, it's learning how to make a good dog, or a good cat. Fine, I'm more of a dog person, but okay. And then, you know, on this side, it's learning how to decompress the instructions in the genome to make that thing, right? To encode the elements of development on this side. So that's the idea as it occurred to us. And then the question is, does this idea have any merit? And can we actually push on it to see, first of all, does it hold up to what we know about the way that the genome actually encodes the form of the organism? And then does it give us anything else out that we didn't put in already, right? Because then once you're at that point, if it's giving you something extra, you can be confident maybe that there's something to it, and that feels like there might be. So the idea here is that the genome instantiates a generative model, which is this sort of distributed, embodied set of information that is used to generate a new instance of the organism. And in that case, in that sense, it's not just a metaphor, it's like directly what the term generative model means. Just by definition, that is what it is. But the properties of it are what make it interesting. So it's an informational bottleneck, as I said before, and the latent variables in the DNA sequence comprise this compressed representation given, it's a representation of the organism only given a decoder that knows how to interpret it. And the trick is that the genome encodes its own decoder. In fact, it encodes lots of its own decoders as it goes along, right? So the genome in the fertilized egg is enough to kick off that process of interpretation, and then it gets more complicated as we go along. Okay, so what are these latent variables in the genome? We know what they are in an artificial neural network. In the genome, basically it's the same idea. It's elements in the gene regulatory network. So the elements there are the DNA encoding for proteins or functional RNAs that may be regulatory, and also encoding binding sites, regulatory elements for those proteins to bind to. And together, the relative affinities of those things are like weights in a network. And that means you can think about the genomic encoding using network science, using dynamical systems theory and so on, things that we don't often formalize in developmental biology, which I think can be really actually productive. So if we think about these weights, what they actually are, when we think about what a gene is, of course, it has this blueprint, as Jacob and Monod said, for a particular protein in this case, but it also has regulatory elements bound by other regulatory proteins, right? So it's the binding of these proteins to the DNA that encodes the weights that I'm talking about. And those weights can change. They can be changed by evolution by mutating the sequence of the binding sites or the proteins themselves. So if you think about gene regulatory networks, people have often kind of zoomed in on them and look, what's a gene regulatory network in a particular organism, in a particular tissue. This is an example from a sea urchin where it's been really well worked out, the developmental logic of sort of these binary self-ate decisions, where you can say, here's an example gene A, and it has a coding sequence, but it also has these regulatory elements, these little pig bits. And if you expand one of those, then what you can see, it's got binding sites for its own product, right, for protein A, but also for protein B. And protein B is a product of gene B, gene A regulates gene B. So you can build up this kind of a network where you can get a positive loop like this, you get positive feedback or negative feedback, and then all these sort of interactions that will ultimately act like switches, where something goes from one, switches into one pathway versus another and activates one differentiation program versus another. And that's all fine. It's a sort of a Boolean logic view where each regulatory element of the gene is acting like an AND gate or an OR gate. So it's doing a logical operation. It's like if protein A and B are here, turn on C. And then it's a series of switches like that. And it's nice and interpretable, right? The problem is it's only interpretable because we've put only 6 genes on here, right? And in fact, of course, you've got whatever, 20,000 genes all doing this at the same time in parallel. So it's, and with massive kind of more complex interactions. So if you zoom out a little bit, rather than thinking about these things as all or none logical switches, which are just doing their job in an abstract space, you have to think, well, no, this is actually happening in real dynamical time, all at the same time, across the whole embryo or across the whole genome in every cell. And so a zoomed out dynamical systems view can be more useful, where you can build a network like this with all these different interactions, and then say, given some input, this network will have excess energy and will find a low energy stable state of gene expression of all these different genes. Okay? And so it'll generate this kind of a tractor landscape, which I'll say more about in a minute. So the idea is here, rather than thinking about one gene at a time, think about all of the genes in every cell and the profile of gene expression in every cell. All, say, 20,000 genes, which are they up or down, not just on or off, but more or less, right? So it's analog, it's not digital. So you can think about the system, the whole system state with genes on or off, more or less. You can do this for neurons, neuronal states as well. And then you can think about the interactions between components as being activating or repressing, all of that generating this kind of energy tension which has to be resolved. So what you end up with is that some combinations of states are stable and some are not. So here's a simple example. Imagine we have just 2 genes, A and B, that can regulate each other. And we have a scenario here where they don't regulate each other, right? They're independent. Then all 4 of these states are stable. They could both be on, both off, one on, one off, right? If they activate each other like this, then only both on or both off are stable. You can't have just one on and the other off because it'll turn the other one on, right? So now we have taken 4 possibilities and restricted it to 2. Only 2 of the possibilities are stable. This is another case where they're inhibiting each other, in which case both on can't be stable, which is an interesting scenario because there you can get a kind of a switch, a competition between them. You can get a dynamic where if they're regulating each other, then you can draw this kind of energy landscape where the high pieces of the landscape are unstable. It can't stay in that state because it's not stable. One of them will start to win, and when that happens, it'll drive it to, making loads of B and not much A, or loads of A and not much B. Okay. So that's just for 2 genes. Of course, you can imagine for all of the genes, like in our genome, there's about 3,000 transcription factors who are just, their job is just regulating all the other genes, right? So you end up with these multi-dimensional kind of states, which you can summarize in this kind of a way, but this is really, it should be a 20,000 dimensional space. The point is that it has stable states that the system can be in, and then loads of state space that it just never is in for any length of time, right? And those stable states that are what we recognize as cell types, right? That's a liver cell, or a glutamatergic neuron, or an epidermal cell, right? That's a stable profile of gene expression in a dynamical systems view. So we can look to dynamical systems theories then for some mathematical methods, right? There's tons of stuff worked out for these kinds of things that think about their behavior and what pushes them into various states, and also how they evolve through time. Because of course, what's interesting in development is you don't just have the embryo suddenly adopting all these stable states and just sitting there. It goes through trajectories of states, each of which is actually temporarily stable, but then gets unstable and causes a further differentiation to happen. So you can also map kind of trajectories through state space and time, which give a better kind of a view of developmental trajectories. And this harkens back to things that you guys may be thinking of, which is Conrad Waddington's early work on what he called the epigenetic landscape. And he meant epigenetic in the referring to epigenesis, right, Aristotle's term, not referring to like chromatin biology and so on, which is how it's come to be used. Although as it happens, chromatin biology is one of the means of regulation, but it's not the only one. So his idea was that you could have this landscape, you could represent development as this landscape, where these channels or canals in the landscape are the stable states that this little ball, a cell in the embryo, can find itself in. Right? And it won't stay on these ridges, which are unstable. It'll end up in either cell type 1, 2, 3, or 4. Right? So for any given cell, what was interesting in Waddington's view was that actually it wasn't deterministic. You could have a little bit of noise. So when the cell came down and hit this first bifurcation point, it might go left or right. And the shape of the landscape would determine, statistically speaking, what proportion went left or right. So how much nervous system tissue you make versus epidermis when your neurectoderm is differentiating. But that was a statistical outcome, right? So it's the law of large numbers here is giving robustness to the developmental program rather than trying to micromanage every single cell's activity at every moment. Now, this view is really well known. That's sort of this famous visual metaphor from Waddington. What's less well known is what he had underneath there, which is, so this is a metaphor of its time. His idea was this, the landscape was like a canvas, and underneath it there were these ropes pulling down on it, like like making like a circus tent kind of thing, right? With pegs that were pegged into the ground. So each of these is a little peg, and you can think of those as the genes that are pulling on the landscape. But really, really collectively. I mean, this is just an amazingly distributed kind of encoding of the shape of the landscape, not due to any one gene's activity at any place. Very much diffuse, distributed kind of encoding. And it turns out that when you work out the mathematics of the encoding within these neural networks, it generates something that really looks like these landscapes, right? It generates these energy landscapes that mathematically are basically equivalent to the much more vague idea that Waddington had. Okay, so that's the general idea, but then we wanted to think a little bit, okay, well, how does this actually work, right? So on the decoder side, on the developmental side, the idea is that, so what we don't have in real biological systems is all these layers exist at the same time. In the way that they do in this encoder system. But the idea is that the layers emerge sequentially, right? So that actually, as you're going through differentiation, each cell division generates a layer like this, right? The information is being passed from one cell type through these other cell types to effectively decompress it to the final decision. And the idea there would be that actually what the genome is doing is multiplexing cellular states. It encodes many, many possible cellular states in this one-dimensional string of nucleotides. Right? In this compressed representation with lots of possible stable outcomes, as I said, and it encodes the nature of their trajectories, and it does that because each cell type inherits a new model. because the model is epigenetically marked, actually. When cells divide, there's chromatin marks and there's also transcription factors and other things that the cells themselves inherit. So the model is different and the decoder itself is actually different for each cell type. So that's what determines, in a sense, why, when you have one lineage developing, that its possible outcomes are restricted as they go through development. So the argument then is that patterning and cellular differentiation, emerge from the genome encoding these gene regulatory networks, which establish these attractor states and trajectories, which constrain gene expression. And I think that word is important, right? They're not driving it. The genome is not actively doing anything. You shouldn't think of gene action in the sort of classical way. It's a set of constraints which make it more likely that the cell will adopt some states versus others. And it's not having to actively do stuff, because the doing is done by all the matter that's there, right? All of the physics, the properties of the proteins that are made is just going to drive what happens. It's constrained by the information in the genome, but it doesn't have to be done by the genome. And I think that's an important distinction, because some people object to the idea of genes being drivers, and I think they're right to object to it. It doesn't have to be. The genome just doesn't have to encode all that stuff. Physics will do the trick, right? So it doesn't encode like the protein folding. The genome doesn't encode that. It just encodes the sequence. And hydrophobicity and charge interactions specify the coding. Now, one of the things that's really important here, as I said, the problem with the blueprint metaphor and the program metaphor in an algorithmic sense is they're very deterministic. Right? It's like you're always going to get the same outcome. It's going to follow exactly the same steps. All the information is there in the thing. It doesn't matter what's outside it. Which just is not the way development happens. So development happens in a more interactive kind of a way, constrained by what's in the genome, but in a kind of a dialogue, in some cases, with environmental conditions and so on. And it also is just variable. You can run the same program twice, and you won't get exactly the same outcome. And if you think about that variability in these artificial neural nets, they have to add it. So variational autoencoders, for example, add variability, and I'll show you how they do that. So this is a traditional autoencoder where its job was to learn to recognize a bunch of images of things. It started out with like digits on envelopes, like zip codes, so it could read those automatically. And then it could take a degraded kind of an image, and if it had learned about those things before, then it could regenerate the image. But the idea was that the reconstructed input would be exactly the same as what the real input was, even if it only got a bit of a degraded one, right? But actually, if you wanted to generate a new thing, like when I made a new horse that had never been made before, then you actually have to add some variability. And the way that they do that is that these latent state distributions are not single weights, they're distributions. Each weight is a distribution. And every time something has to pass through one of the weights, the distribution is sampled, and a random element value from that distribution is used. Right? So it adds noise, and it encodes. In fact, it encodes the variance as well within there. Right? We don't have to do that. Biology doesn't have to do that, because everything that biology is made of is really noisy. Just physically speaking, molecules just are jittering around. They're subject to thermal fluctuations. They're subject to quantum noise in some senses, and so everything that's happening in development is already variable. And in fact, the challenge for evolution is to find robust encodings that can deal with all of that noise and still make a viable offspring at the end of it. So when you go from genotype to phenotype, you're always going to get some spread of outcomes. Doesn't matter how tightly you try to encode things. And that becomes really important, because evolution has to deal with noisy components. Like at any moment, you could say, okay, I need this transcription factor to be on or off. Well, sometimes it's going to be making 15 molecules of protein, and sometimes it's going to be making 25 molecules of protein. It's just going to vary like that, and it's small numbers. So the gene regulatory networks have to be able to cope with that noise. And a consequence of that is that they can cope with some genetic variation. Right? So a little bit of genetic variation in those regulatory elements that are saying how much of each protein to make it can already cope with a little variation in those parameters. What that means is that genetic variation can build up in the population. It's not so deterministic that it's over-specced in an engineering sense. It can't deal with any noise. So the fact that it's robust is the very thing that makes it evolvable. It sounds kind of paradoxical, but it's because it allows evolution to accumulate. So if we think then on the evolutionary side, how does the encoder work? The argument here is that in a very direct way, evolution is acting like a learning process which is setting the weights of a gene regulatory network, really analogous to an artificial neural network, or indeed to the way that we learn, through Hebbian learning and synaptic plasticity and so on. Of course, evolution doesn't do that in the same way that learning algorithms do in artificial systems. It does it with a different algorithm, but the effect is the same. It evaluates it based on the outcome, and then it selects the genotypes that are most successful. And what it does is do that in parallel over the whole population. That becomes really important, quite different from artificial neural networks. Okay, but I think the effect is the same. This is a really interesting paper which goes into that idea. Okay, so what does this way of thinking get us? Well, the properties of the generative model, as I said, this compressed representation with a distributed coding of phenotypes. I think those are just accurate ways of describing how the genotype relates to the phenotypes, really indirect for the most part. Doesn't mean you can't find occasional one-to-one gene mappings, but those are exceptions. And in a way, it's unfortunate that Mendel started with really clear single gene examples because it polluted everybody's mind into thinking along those lines. And actually, most genetic coding is just massively, massively polygenic. It also, this way of thinking explains developmental variability and robustness and evolvability. And those are all hard to deal with in a blueprint metaphor or even a program metaphor. It's like, okay, you can call it that for this instance right now, but how did it get that way and how can it change? It's not, those metaphors just don't lend themselves well to the fluid, evolvable nature of these kinds of encodings. So just a few examples. First of all, we were talking earlier how a lot of times in science, we like to tell nice, clean stories, right? So if we're thinking about some development of something, right? Development of the telencephalon, and there's a gene here, sonic hedgehog, which is responsible for patterning the midline and splitting the development telencephalon into two halves, right? And we know that because when it's not there, you just get one. Okay? Nice, clean story, single gene, it's all good, right? Of course, it's a fantasy because it's working with loads of other genes there doing this job in this context, and the same gene is doing something completely different in the limb, right? Patterning the thumb to pinky axis in the limb in conjunction with other proteins in a different context. And in the developing spinal cord, it's both patterning progenitors and it's acting as an axon guidance molecule. So every time we have some nice developmental story, it's always just incredibly specific and not saying actually what that gene does overall, and not saying the gene does that by itself because it's never doing it by itself. Right? So we can have a much more distributed kind of view of the encoding. How you encode a limb is a lot more than just having sonic hedgehog more over here than over here. Right? And this becomes really important when we think about genetic variation. As I said, Mendel's traits were really exceptional. Most traits are really, really polygenic. And we're learning this especially from human genetics, looking for the genetic basis of all kinds of common traits, but also risk for various common disorders, where there's just thousands and thousands of genetic variants that can affect things like risk for schizophrenia, say. And on the flip side, most single variants are pleiotropic, which means most variants that people find in the genome don't just map to one trait, they map to many traits. Okay, so we have a many-to-many mapping in both directions, right? It's not only not one directional this way, it's not one directional the other way either. And so first of all, in human genetics, people just use linear additive models. They just think about the burden of risk variance, say for schizophrenia, right? As opposed to combinations which are pushing you into various risk domains. It's just easier to do that. It's an idealization that's mathematically convenient, And it's fine once you realize that you're making an idealization and you don't think that that's how the biology actually works, because it won't be like that, right? It'll be much more nonlinear in its nature, in that mapping. But the other thing about this is a mystery. If most traits are polygenic and most variants are pleiotropic, how can you ever get selection on any trait? Because every set of variants that is predisposing to one trait will also be affecting loads of other traits at the same time. And it seems like you should get a kind of a gridlock that, this seems like a problem, right? How can you ever get selection on one set of variants when they're all doing multiple jobs and affecting lots of traits? And here, this is one of the areas where this model, thinking about it from the AI machine learning realm and also from neuroscience, gave us what I think is a really interesting potential insight, which is that this is a problem in neuroscience, for example, in our visual system that learns to characterize elements, say, of people's faces. We can think about those along various dimensions that are separable from each other. And you can see in the logic of representations of different faces in parts of our visual cortex that actually, in a simple sense, it looks like there's so many shared neurons that are representing Joe versus Sam that how could the system possibly tell them apart? But actually, you can see that in a nonlinear encoding, these are in mathematical subspaces. Right? And this is where it gets quite abstract in a mathematical sense, but the brain can distinguish those subspaces because the configuration of the rest of the brain that's looking at these encodings has learned to tell them apart. Okay? Maybe that's a little clearer here. So in these sort of like variational autoencoders, you can get them to make people, right? You can draw people's faces. And they can encode along separate dimensions these different aspects, like the age or the smile or the pose or the gender, right? So within their representation, this compressed representation of faces, it has learned that there are some dimensions along which they tend to vary independently from each other. That's why you can do your face app thing that makes you, when you age your face, it's because this compressed representation has these different subspaces within it that are abstract in this nonlinear space. So the argument that we're making is that something like that is going to happen for the representations of different traits. They are highly nonlinear and they'll be pushed into different subspaces of that latent variable space. And especially that's true if there has been past volatility of the environment, which has tended to put some selective pressure on different elements at different times. Then the system learns to manipulate them independently, basically. So you can think, rather than having this sort of thing where all these pleiotropic genetic variants are so tied up with each other that they can't be separated, if you're just in a simple encoding, that's true, but that's not the nature of the encoding. It really is this nonlinear thing where these become separate traits that can be pushed in these latent spaces without affecting the other traits. This is one last thing that I think is really interesting, and it's something that I actually only learned about recently, but it's interesting because there's another parallel to particularly neural representations in neural networks. This is called developmental systems drift, and the idea here is that you can have one genetic regulatory network that codes for one phenotype. So here's just a species genome codes for its phenotype. And you can get like genetic drift, where elements of that network are changing, and you get a concomitant change in the phenotype, gradually, just at random chance. Okay. Or you can get like strong selection, which can really drive that one network to a different one, and now it's making a different phenotype, right, for a reason, because this has been selected for. That's all fine. What's really interesting is you can get this phenomenon where the genetic regulatory element changes in ways that don't change the phenotype. Okay? Because it's a robust encoding, you could get a few little mutations that the system can tolerate. Every time you get one of those mutations, it changes the landscape that can now be tolerated. and it changes it again and again and again. So you can gradually drift away from the original encoding in this network while maintaining the phenotype. And you can see that this is the case because it can get drift in different ways, right? Where you could have two species that are both making the same phenotype, and when you hybridize them together, now they don't work. These two genetic networks are now incompatible with each other. So you get a kind of a hybrid incompatibility where the hybrids, both of the parents have the same phenotype, and now the hybrids have something else. So there's a few really interesting examples of that. This is one from Drosophila. This is a Drosophila melanogaster showing the bristles on the thorax, and Drosophila simulans looks exactly the same as that, but the hybrid looks like this. It's got all weird numbers of bristles. Right? So both of them can make the same program, but the hybrid can't. And to me, that also fits well with this generative model encoding idea. It's much harder to fit with the other sorts of ideas. So the idea is you get these different sorts of shift in the weightings of the network, but the phenotype stays the same. What's interesting there is, again, there's a potentially direct analogy to what's called representational drift in neuroscience, where, for example, people have found, like, if you're encoding, like, from the visual cortex of a mouse, say, with lots of electrodes, you can see a pattern that represents something, that some stimulus in the environment. Over time, that pattern can change. The individual neurons become less or more active, but the overall pattern still means to the mouse X is out in the world, and you can tell because it behaves like X is out in the world. Right? So the representation drifts because there's some synaptic plasticity, there's some ongoing noise and change, but the system as a whole maintains what the pattern means while the individual elements are allowed to change. And I think you could see the same thing happening in a genetic network. There's enough play in the system that it could drift while there's some pressure to keep the phenotype the same. So, so in terms of then thinking about Evo-Devo, which is the sort of last point I want to make, I think, again, here we're fond of these neat stories where, like, the one gene that's responsible for this one difference in these phenotypes, hopeful monsters, as we were talking about earlier. This is a good example where it's a really lovely story. It happens to be a brilliant story because in the beaks of Darwin's finches, and they found out that the levels of this protein, bone morphogenetic protein 4, were different in the anlagen of the beaks, that in a way that seemed to explain the size of the beak that emerges. So beautiful, nice and neat. The question is whether this really is a likely or typical scenario for how evolution actually happens. Right? Does it happen that you have an animal with one kind of beak, and suddenly there's some big mutation in BMP4, and now it's got a big beak? It just seems like not a very likely way for evolution to happen to me. I think more likely is that you get this gradual change along these dimensions that are encoded in separate subspaces in the genome, which might then eventually get locked in later on by a large effect mutation. But it seems less likely that the initiating event would be this one-off thing where suddenly small-beaked birds have a large-beaked baby. Yeah. Okay. Just the last 2 seconds then. The question is, like, whether this is more than just a metaphor, and I think it can be, although I think we still need to really push on it a lot more, but I think it can be productive, and people are using it, they're not using it, they have been using approaches that I think are useful here, and one of them is to look at real systems biology data, where now we can actually get, so like single cell RNA sequencing from every cell in a developing embryo, so you can really see what that gene profile looks like. You can start to trace trajectories. You can start to look at the effects of a signal like Sonic Hedgehog on, developing progenitors in the spinal cord, which is what this paper is about from James Briscoe, who's done a lot of this, where, they're really using the mathematical tools of dynamical systems to model their empirical data, and they're finding that it really does match this view of attractor states and stuff that Waddington had already described in a conceptual way, 70, 80 years ago. So I find this really exciting, because what it means is that, the problem with Waddington's metaphor was it was I don't think anybody thought it was wrong. It just wasn't actionable. They just couldn't do anything with it because all they could do was just like modulate one gene at a time or track 2 or 3 genes at a time. But now we can do holistic get holistic data. We need the sort of holistic computational models and concepts, I think, to help us make sense of it. And in fact, this is a paper actually using variational autoencoders to learn the structure of genomic regulatory networks, and it works really well. And I think it works really well because it actually matches the structure of gene regulatory networks. Like there's a natural sort of a match there. The last thing that we're thinking of doing, myself and Nick Cheney, is making these artificial virtual robot-y type things, artificial life systems, these little squishy robots that can move around in a virtual environment. I'm thinking about using this. So right now they have a kind of a direct encoding where X encodes the red bits and Y encodes the blue bits and so on. And what we want to do is actually take a step back and make it an indirect encoding, and then that could be more evolvable in time, because this is not very evolvable. That's the problem. Okay, so just to summarize then, our argument is that the genome instantiates a generative model of the organism. As I said, generative model in the broadest sense, I think that's just an accurate statement. I think that's just what it is. But whether the implications of that from our machine learning and so on are relevant, I think is open to empirical testing. But our argument would be that the genome is this compressed distributed representation in this latent variable space that evolution learns. And its job is to constrain the self-organizing processes of development. They're going to do something. What it does is make them do dog instead of doing cat. So shaping the organismal form. And I didn't talk about the nature, but the idea is that the nature of the organism, the dogginess or the cattiness, just comes from shaping their brains, basically, into dog brains or cat brains. Okay, great. I'll leave it there. Thank you very much.